everyone, and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to study about microbial growth, that is part 2. So in the previous lecture we have discussed about the microbial growth in detail. We have discussed about the factors that are affecting the growth of microbes. We have seen the bacterial growth curve. We have seen the different phases of bacterial growth curve, like how the bacteria grow, and what does the lag phase is all about. How does bacteria prepare themselves in the lag phase? We have seen that how bacteria get the exponential growth in the log phase. What is a stationary phase? What is the decline or death phase? And some questions related to the topic. So today in this lecture we will going to study about the part 2 of microbial growth. So measuring numbers of microbes. So now we have seen how bacteria has growing, how microbes are using the favorable conditions, using the nutrients and using the moisture content for their growth. Now after growing, like we all know that bacteria can double themselves within a half an hour, or the maximum time is 60 minutes, or we have seen the shortest time is 10 to 20 minutes. So within 10 to 20 minutes bacteria double itself, means a single bacteria, they divide itself and forms a two new daughter cell that are equally, or you can say that are identical to each other, with each other. So in this particular term, that is measuring the number of microbes, then it is very difficult to measure the number of microbes, isn't it? So there are some different methods through which you can measure the microbes. The first one is indirect measurement. So in under the indirect measurement, measures a property of the mass of cell and then estimate the number of microbes. So in this particular term, that is indirect measurement, so under this we what in this method what it is done, the property of the mass cell, the mass cell is measured, cell of the mass of any cell is measured, and then after measuring the mass of the cell, then you estimate the number of the microbes. Then after measuring, then you estimate the number of microbes. Then comes the turbidity. Turbidity can hold tube up to the light and look for the cloudiness as evidence of growth. That is difficult to detect slight growth. The spectrophotometer can measure how much light a solution of microbial cell transmit. The greater the mass of the cell in the culture, the greater its turbidity, that is known as cloudiness, and the less light will be transmitted. So what is the advantages of this turbidity? That no sensitive in terms of numbers of bacteria cell and not useful for detecting minor contamination. So turbidity is nothing. If you ever define turbidity, so turbidity is defined as the cloudiness, the dhundlapan, you can say. And they can hold a tube up to the light, and means you are holding, like this is a test tube. So this is a test tube. So this you want, what you do under the to check the turbidity, that you hold this test tube against the light. Then a spectrophotometer can measure, means if you talk about the spectrophotometer, they can measure how much light a solution of microbe cell transmits. So any spectrophotometer, since what it is doing, so it is using a light, light, and through that light they can measure that how much light a solution, means a solution having a microbes that how much light a solution of a microbial cell transmit, means a solution that is full of microbe cell, then how much light can they transmit is measured through a spectrophotometer. So it says the greater the number of cell in the culture, the greater will be the turbidity, or you can say the cloudiness. So what is the disadvantage? So no sensitive in terms of number of bacteria cell and not useful for detecting minor contamination. If you have a very minor contamination, so those contamination cannot be detected. Then comes the metabolic activities. So we all know that this metabolic activity take place through three ways. The first one is the rate of formation of metabolic product such as gas or acid that culture produces. Second is the rate of utilization of substrate such as oxygen or glucose, and the last is the rate of reduction of certain dyes, that is example methylene blue becomes colorless when reduced. So metabolic activities are of three types. The first one is says the rate of formation of metabolic rates product. Metabolic product means how a metabolic product is formed, such as gas, such as acid, that that have a culture produce. Then comes the rate of utilization of substrates such as oxygen or glucose. That is how the substrate is utilized, like such as oxygen, such as glucose. These all are act as a substrate. Then comes the reduction of certain dyes, how to reduce certain dyes, like for example methylene blue becomes colorless when reduced. So these are three metabolic activities that possess in three ways. Now after completing the indirect measurement, that how indirect measurement measure the number of microbes, then comes the direct measurement. So direct measurement gives some more accurate number of microbes. Since this method is very direct, so this method gives a very you can say a direct number of microbes. They measures the number of microbes very accurately, you can say very accurate, or you can say as compared to indirect measurement. This direct measurement this measures the microbes very accurately. So under the indirect measurement, it comes the direct count, second is plate count, third is second is plate count, and fourth last one is filtration. So if we check the direct count, then qualitative control, electronic counter, rapid and accurate only if bacteria cells are only particles present in the solution. So direct count is a type of a count where you directly what you do, it is a very rapid and it is a very accurate only if the bacteria cells are the particles present in the solution. Means if you have a solution and under that and inside that solution you have the bacteria cells, that is in the form of, you can say, a particle form. So the direct count gives a very accurate and the very rapid, means very jaldi wala count. So it gives the total count of life plus as well as death, means how many bacteria cells are live and how many bacteria cells are dead. So it gives both the type of a count. Then comes the plate count. Under the plate count, bacteria colonies are viewed through a magnifying glass against the colony counting grid called a cubic colony count. That gives the viable count. Then comes the filtration. So under filtration, a known volume of liquid or air is drawn drawn through a cell membrane filter by vacuum. The pores in the filter are too small for microbial cell to pass through. Then the filter is placed on the appropriate solid medium and incubated. The number of colonies that develop is the number of viable microbial cell in the volume of liquid that was filtered. So this technique was great for concentrating a sample, example a swimming pool where the small population may go undetected using some other method. So what is filtration? So filtration is another technique through which you can measure the microbes in this a very known volume of liquid, or you can say air is drawn through a membrane filter using a vacuum. So the pores in the filter are too small for microbial cell to pass through it. Means, see, you are using a filter, and filter is having a pores that are so small that a small amount of small number of microbes, even a very tiny microbes, cannot pass through it. So then the filter is placed on the very appropriate solid and medium, and then you can incubate, and the number of colonies that are developed is the number of the viable cell, right? So in this it gives the viable count only. Now coming to the next, that is the growth factors. So as far as the measure, how you measure the number of microbe is concerned, then comes the growth factors. So microbes can exist in a great many environment because they are small, easily dispersed, need only small quantities of nutrients, are diverse in their nutritional requirements. So whenever it is considered concerned with a growth factor, so bacteria needs, not bacteria but every microorganism needs certain factors for their proper growth, right? Like mainly physical and chemical factors are there. So under the physical factor, pH, bacteria can be classified as acidophiles, neutrophiles, alkaliphiles. So acidophiles is the acid loving, that is grow best at the pH of 1 to 5.4, that is lactobacillus. Then comes the neutrophiles that exist from a pH to 5.4 to 8.5, most bacteria that cause human diseases. And last is the alkalophiles, that is a base loving bacteria, and the pH range from 7 to 11.5, that is vibrio cholera. So according under the heading of pH, so bacteria can be classified into three categories. The first is acidophiles. Since its name is acidophiles, so it is a acid loving bacteria. So the optimum pH for their growth is 1 to 5.4. All the lactobacillus species are acidophiles or acid living. Then comes the neutrophiles. So neutrophiles under this are very bacteria that has a very neutral pH, that is 5.4 to 8.5, can exist in this category, and they causes human diseases. Then comes the alkaliphiles. Alkali means they are the base loving, and the pH is 7 to 11.5, example vibrio cholera, that causes cholera. So all these type of bacteria have their categories under the and according to the pH they are classified as acido, neutro or alkaliphiles. Then comes the temperature. So comes the temperature. So bacteria can be classified as psychrophiles, mesophiles and thermophiles. So under the heading of psychrophiles, so psychro means they are cold loving bacteria. So they can grow easily around the temperature of 0 degree centigrade, and the optimum temperature is 15 to 20 degrees centigrade. Then comes the mesophiles. So they can best grow in 25 to 40 degrees centigrade. a human body temperature is 37 degrees centigrade. So mesophiles can grow easily on in the room temperature. Then comes the thermophiles. They are the heat loving bacteria. They found between 50 to 60 degrees centigrade, and found in the compost heap and boiling hot spring. Then after completing this, let's move to the moisture. We all know that any bacteria or any microbes can easily grow where there is a abundant amount of moisture. So whenever moisture is concerned, bacteria can easily grow in moisture. So only the spore of the spore forming bacteria can exist in the dormant state in a dry environment. So there are numbers of bacteria, or you can say microbes, that found moisture or water as a very favorable condition for their growth. So in this way, moisture play an important role in the growth of any microbes. Then after coming to the moisture, then comes the hydrostatic pressure. So this hydrostatic pressure is exerted by standing water, example lakes and ocean. Some bacteria can only survive in high hydrostatic pressure environment. The high pressure is necessary to keep their enzymes in proper 3D shape. Without it, if the enzyme lose their shape and denature, and cell dies. So hydrostatic pressure is a pressure that is exerted by any standing water, like any water, like lakes or ocean. So here some bacteria can only survive in the high hydrostatic pressure, while others die, because there are certain bacteria that can you can withstand this high hydrostatic pressure. Then comes the tonicity. Tonicity is hypertonic, hypotonic and isotonic. The use of salt as preservative is curing meat, and the use of sugar in making jellies is based on the fact that hypertonic environment kills or inhibit the microbial growth. Halophiles means salt lovers inhibit the ocean. So hypotonic or hypertonic and isotonic. So the use of salt is whenever you are using the salt, so salt act as a preservative, and these salt is help in curing the meat, and this sugar is also used as a preservative. And you, by using all these preservative, what are you doing? You are just inhibiting the growth of any microbes growth. Halophiles are those microbes that has a salt that is a salt lovers, means they can easily survive where the concentration of salt is high, like ocean water, lake water. Then comes the radiation. UV rays and gamma rays can cause mutation in DNA and even kills the microorganism. Some bacteria have enzyme system and can repair some mutations. So what is radiation? So UV rays and gamma rays can cause mutation in DNA. We all know that whenever it's a UV or a gamma rays, so they are causing mutation in DNA, and even they are killing the microorganism. That is why a laminar airflow is before starting your experiment in under a laminar airflow, UV the lights are switched on just to kill, or you can say inhibit, the growth of microorganism. Some bacteria have enzyme system that can repair some mutation. Now comes the oxygen requirement. Whenever it's a hydrogen oxygen requirement, strict or obligate anaerobes, oxygen kills the bacteria, example Clostridium. Strict or obligate aerobes, that has a lack of oxygen and kills bacteria. Then comes the facultative anaerobes that can shift their mutation metabolism, example E. coli. Then comes the aerotolerant, the bacteria that don't use oxygen, but oxygen doesn't harm them. Example, lactobacillus. Then comes the micro aerophiles. They like low oxygen concentration and higher carbon dioxide concentration. Example, campylobacter bacteria. So these are certain different types of oxygen, uh, uh, under the very low oxygen. Some bacteria can grow, some bacteria cannot tolerate the use of oxygen, but the oxygen they cannot harm them. So these are certain microbes that according to their you can say machinery, according to their characteristic, they can easily grow. Then comes the nutritional factor. Nutrient needed by microorganism includes carbon, nitrogen, sulfur, phosphorus, vitamins, certain trace elements. So whenever it's a carbon, then carbon containing compounds are needed as an energy source, for example glucose for building blocks. Then comes nitrogen. They are needed for amino acid and nucleotide. Some can synthesize all 20 amino acid, and uh, other have to have some provided in their medium. Then comes the sulfur. So they are needed for amino acid, that is coenzyme. Then comes the phosphorus. They needed ATP, phospholipids and nucleotides. Vitamins. Vitamin is an organic substance that an organism requires in a small amount, and that is typically used as coenzyme. Many bacteria make their own, but some are required in the medium. Microbes living in the human intestine manufacture vitamin K needed for the blood clotting and some of the B vitamin, thus benefiting their host. Certain trace element, example copper, iron, zinc, sodium, chloride, potassium, calcium, etc., often serve as a cofactor in enzymatic reactions. So again, nutritional that has a biochemical factors are also needed by microorganisms for their proper growth. So carbon, since they use glucose as a source of their building blocks, they needs oxygen. They are nitrogen. They are needed for amino acid, or you can say the nucleotide. So there are 20 amino acids, and they all need all these nutritional factors for their proper growth and for their development. So this is about the nutrition, oxygen. So as far as this lecture is concerned, we have covered the how you measure the number of microbes, what is a direct method, what is a indirect method. We have seen the growth factors, how microorganism grow, how they use physical factors like pH, temperature, moisture, hydrostatic pressure, tonicity, radiation. Then how they utilize oxygen requirements, how they utilize the nutritional factors, or you can say biochemical factors, how these factors helps in their proper growth. Now let's quickly discuss the important questions that can be asked in the examination. So the first one says, what are the four phases of bacterial growth curve? So in the part 1 we have discussed the four phases of bacterial growth curve. Then comes, what does the bacteria growth curve show? So what does this growth curve show? You have to go through the part 1 of this topic, and you can easily get answers from there. What are the things bacteria need to grow? Means what are the physical factors, or you can say what are the chemical factors, even how the nutrition play an important role for any bacteria to grow. Then comes how is growth measured in a microbial population, means how the growth of bacteria is measured in any microbial population. And the last one says, how do eukaryotes and bacteria, that is archaea, differ in their reproductive method? So this question number 5 You can for this question you can refer the part 1 of this topic, and from where you get the exact answer. Again, if you have any doubt related to any question or any slide you want me to repeat, so you can leave your questions on the comment section below. In the next lecture I'll be answering all your queries. So thank you students for watching. Please do like, share and subscribe. So this is our helpline number, that is 7080833450, and you can visit our website, that is www.itlsacademy.com, to get new update on the topic. Thank you.